I know a lot of you are probably wondering, why is he starting this video eating a piece of cake? Well, it's because today we're going to go over specifically what happens if I eat too much of this cake. So let's get after it. And I'm Dr. H, and I'm the one that will be taking you in this particular lecture. So the reason why I'm wearing pink today is because this is Cancer Awareness Month. So please, when you have the opportunity or you have loved ones, please go get them screened, because the earlier you diagnose this disease, the better the outcome is. Glycogenesis and as well as glycogenolysis. So now someone can come up with a question like, and the simple explanation to this is simple and just based from the words itself. So it's glycogenesis. So glycogenesis, genesis meaning the beginning of something. So this is the beginning of the formation of glycogen. And then glycogenolysis, this lysis part, so meaning we're splitting up the same glycogen molecule. So now, let's go into more detail about glycogenesis. Okay, so guys, we're going to do glycogenesis today, and where I got this idea from is because someone suggested it to me, like they wanted a video on glycogenesis. So if you want a particular video on a particular topic in biochemistry or any other course that you might feel you need help in, just feel free to comment down below or just comment on our various social media platforms so that you can get this content to you for free. So now let's start with the glycogenesis. So for glycogenesis, like I said in the beginning of the video, glycogenesis, this happens when you're eating too much glucose, when you're eating too much carbohydrates, when you're eating too much cake, you're eating too much bread. It's too much for you now. Your body now wants to store it. So again, we know from the previous, like I said, when you break down starch, when you break down all the carbohydrates from the food that you're eating, the end product that we want is glucose. So when you eat, your what is known as the fed state, and when you're in the fed state, there's a lot of glucose in your body. So the glucose will now enter your cells. So the thing is, when you have a lot of glucose in your body, insulin gets released, and when insulin is released, it will facilitate the entry of glucose inside the cell. So now what happens inside the cell when glucose enters, for it to be converted into a form that you can store and use for later use? So the thing is that this is glucose. So again, glucose, when we're looking at glucose, it has 6 carbons. So this carbon number 1, 2, 3, 4, 5, and 6 here, right? So this is glucose. And then again, you're seeing a lot of cartoons. It's probably confusing at this point in time, but I want it to make sense for you as we go on further in the lecture. So this is a happy process. The glucose is happy to be in the cell. It is no longer happy to just be floating around in the blood, which we don't want. So then again, so when it enters, the first reaction that happens will be catalyzed by these two guys. If you've watched our previous video on glycolysis, you understand and remember these two guys. So this guy over here, again, I draw him to be sort of showing like the muscles, because the thing is that it is predominantly found in the muscle cells. So this guy is what is known as hexokinase, and this guy is glucokinase. So what do these guys do? So here's the thing. So what these guys will do is there will be a molecule of ATP floating around, and then again ATP 1, 2, 3. So one phosphate, second phosphate, and third phosphate here. So it'll grab onto a phosphate here. And what does it do when it grabs onto this phosphate? So it will put this phosphate here. And what did we say again? This is carbon number 6. Again, 1, 2, 3, 4, 5, 6. So this is where our phosphate will be. So how do we name a glucose molecule that has a phosphate attached to the carbon number 6? It's simple. It will simply be called glucose 6-phosphate. Again, the 6 just telling you that it is attached at the carbon number 6 in the molecule. So now we formed glucose 6-phosphate. Again, too much glucose now gets converted into glucose 6-phosphate. And then again, there's another guy From a previous video, again, if you didn't watch the glycolysis video, I highly recommend you watch it. So I said in the glycolysis video, if you see me drawing an enzyme and I show it having two arms like this, I mean this is sort of serving the function to rearrange the atom into something that we want. But what do we want in this process? Let me show you. So what this guy does, he will grab onto this phosphate here and he'll end up attaching it here. And then again, what did we say? This is carbon number 1. So what will be the name now? So this will be glucose 1 phosphate. Okay, so now reached this point. This guy has rearranged them. This guy's name is phosphohexose mutase. That's the name of the enzyme that's responsible for this reaction over here. So now we have another enzyme here. So I want you to pay particular attention, because up until this point it's been a bit simple, and the process has been pretty straightforward, and I promise it will be even more straightforward. So then again, we have this guy over here wearing the sash, the Kung Fu sash. He looks cool, obviously. He looks like he's shopping something, but what is he shopping in particular? So we have what is known as UTP. So uridine triphosphate. So what this guy will do is that for the UTP as well, so uridine molecule here, so there's phosphate here, another phosphate here, and another one here. So this guy will come and chop straight here. So what products will we have at the end of the day? So when it chops here, these two will live together, and so it will be like this: phosphate and phosphate 2. This is what is known as pyrophosphate. Now you're probably wondering, what is the essence of this reaction? So the thing is that up until this point, this reaction is endogenous, so meaning it is absorbing a lot of energy. Now when we form these two pyrophosphate here, pyrophosphate, again, this phosphate and this phosphate, as it leaves, it will be broken down again by an enzyme known as pyrophosphatase. Now what this means in this reaction is that when it's broken down, this is an exothermic reaction, so meaning it will release energy, and this same energy that will be released from this reaction is the energy that we're going to use subsequently to fuel the rest of the reaction. So this is why this reaction is particularly important. So again, we have pyrophosphate, and now what are we going to be left with? Again, remember, at this point here, at carbon number 1, there's a phosphate attached here, right? Simple. UTP has lost 2 phosphate molecules. What remains here is, sorry, let me use a different color. So what remains is here the uridine base attached on 1 phosphate. But where will we go now? So again, 1 phosphate here. So this will come here. And what will it form? So what it forms now will be what is known as UDP glucose. Again, this guy is a bit upset, and I'll further on explain why I dream in this manner. Yeah, so again, here a carbon atom number 1, so there will be UDP attached, and again we said this is UDP glucose. So in other literature, to simplify things, they say that this UDP glucose is the activated form of glucose. That's why I've put it in this manner to help you understand. So I want you to think of this scenario. So suppose this is an activated form of glucose. I know at some point in your lives you've probably seen someone agitated, someone being angry, and when they're agitated, you kind of want to separate them and just calm the situation down. So the thing is, throughout this whole process, again, you are eating a lot of glucose. This is the fade state. There's a lot of glucose. So this is not the only guy being formed. So there's a lot of these guys being formed. So for simplification's purpose, you just draw them like this. There's a lot of them being formed right now. And then we don't want to have too many of these because they're angry, they're upset in the cell. So what are we going to do about it? This is where these two guys come into the equation. So what these two guys do, and the name of the enzymes is fairly basic. So the first enzyme here is what is known as the branching enzyme. And the second one here is what is known as glycogen synthase. I know you hear the name glycogen synthase and automatically everything is making sense to you because this is the whole process that we're trying to achieve. We're just trying to create more glycogen. So, what do these guys do? These guys do sort of like two different things, but they are complementary to each other because basically we don't want a lot of this guy just existing like this, right? So, what happens is, like I said, we have a lot of these UDP glucose being produced at this point in time, but we don't want any of that to happen. So, to understand what's going to happen here, I want to take you back to the glucose molecule because I want you to understand certain basics about it. That will help you build a deeper connection to what I'm about to explain further. So, when you look at the glucose molecule, again, I said, let me just kind of cut everything else out. Okay, we're focusing solely on the glucose right now. So when you look at glucose, again, 1, 2, 3, 4, 5, 6. So for glucose, it has two sides. There's a reducing side and a non-reducing side. There's a reason why we call them that, and you'll further on understand as we go on further. So in this case, the reducing side is where the carbon number 1 is. So reducing, think of the lowest number, which is 1. So the reducing side, which is where there's the carbon number 1. And the non-reducing side, which is the n here. So we're not going to say the highest number, carbon number 6, no. We're going to say the opposite number from 1, which is 4 here. So at carbon number 4, this is the non-reducing side, right? So now that we understand this concept, there's another important concept that you have to understand. So let's go now into the reaction. So first of all, we're going to talk about glycogen synthase. So what will glycogen synthase do in this reaction to calm down the situation? So that's what this guy will do. And I'm just going to continue from here. So suppose these are his arms. He sees these two guys are fighting and he just wants to calm down the situation. So he'll grab this guy over here and grab this guy over here. So what does he do to them? So he will make them unite. So this will be the bond between them. But there's another important thing you have to take note about these bonds between glucose and glucose. They are known as glycosidic bonds. But there's another important thing you have to take note. So again, here's the thing. So the way the glucose is arranged at this point in time. So at carbon number 1 here, so there's a carbon attached to an OH and an H here, right? Again, so this is carbon number 1 here. This is the carbon number 1. Oh, sorry, this is the carbon number 4 we're talking about here. Carbon number 4. So suppose this other guy here, this carbon number 1, is this guy here. Now, in a situation where this carbon number 1 has an OH here as well, up on top, H down. So when they bond, this is what happens. So this guy, to try and bury the hatchet, will give up both the OH. This guy also decide, okay, you know, I have to sacrifice something for myself as well for the sake of peace. So what does he do? He'll give out the H. So in the end, what will be lost is H2 H2O, or water in short. So the bond that will be here will exist like this. So it will be a carbon. Since there's only this oxygen left here, I've lost the OH and H here, so it will be bonded like this. Oxygen here, bonding these two carbon atoms. So if both of them are on top, the both OH groups are on top, this is what is known as an alpha glycosidic bond. But suppose in a case where, again, this guy had an H up here and the OH was down here. Again, same thing, give out an OH, give out an H, but when they bond in this manner, from top to down, the bond that will be shared will be like this. This is known as a beta glycosidic bond. So the importance of knowing whether it's an alpha or beta glycosidic bond will determine several characteristics about the molecule that will be formed. So for this particular molecule, which is glycogen that we want, it is an alpha bond that is formed always. So this alpha bond is the same bond that amylase will use, because this is a storage form of glucose. So when it's stored, amylase will come in again, break the alpha bond, and then release free glucose. So we'll go into it into detail when I'm talking about glycogenolysis. But in this process, I'm just trying to make you understand. In cellulose, the bond that is there, it is an alpha bond as opposed to, or it's a beta bond as opposed to an alpha bond. So I mean, an amylase will not work on that bond particularly. So this is the reason why we cannot digest food things like grass, because we lack those enzymes to break down those beta bonds. So again, so the bond is between carbon number 1 and 4. And then again, we said alpha glycosidic. So when we're simply describing it, we're going to say this is an alpha 1 4 glycosidic bond, right? I hope this is clear. So again, a lot of these guys have been formed. Again, this guy is being the peacemaker, so he's making multiple bonds again. So here another bond, here another alpha 1 4, here another alpha 1 4, here another one alpha 1 4, here, and everywhere. This guy's face will even change now. We'll be happy, 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 everywhere, happiness everywhere. So now, one thing you have to understand about this whole process is that this cannot continuously go on, because if it's just forming this long chain, eventually the space is bound to finish in itself. So this is why this guy will come in again, because okay, so this guy is also trying to establish the peace, but how will we do it? So again, this guy will come here, grab onto him, and grab onto him. But what kind of bond will be formed between these two? So the bond will be between carbon number 6 here and carbon number 1 here. And again, it will be straight down like this. And then I said again, glycogen, it is an alpha glycosidic bond that's been formed. So the fact that it's between 1 and 6, so this will be a thing. The naming will be an alpha 1 6 glycosidic bond. Okay, so again, if you look at the glycogen molecule the way it is, so again, I don't know if you remember, I'm going to give you 3 seconds to pause to tell me right now which is the reducing end and which one is the non-reducing end. So okay, I knew you guys already knew. So here's the thing. The reducing end, we said, again, reducing, remember, the least number. So this is carbon number 1. So this is the reducing end here. This is the reducing end here. The non-reducing ends are these ones here. So these are the non-reducing ends. So with that, it concludes glycogenesis. I hope it all made sense for you. And if you have any other questions about the same topic, please come through. And we're now going to go into our other installment of glycogenolysis.